Sweet. So we're group 4. I'm Keegan, and I have Tianna and Penny in my group as well. And we're going to start presenting the lesson 9. I'm going to pull up our work so you can follow along, and then we will begin. Okay, and I will start with number 1. So for number 1, it was the diagramming the intermediate stage. Let me go here really quick. Okay. Diagramming intermediate stage of Bacterium E. coli. It said you could do linear. I ended up doing the circular one and keeping it. So the only thing that I forgot to put the RNA primers that would follow the helicase at the where the strands are splitting. And so that would go like right here on both sides at the junctions. And we have everything there. We have the lagging strands. You can see the little Okazaki fragments. You can see the origin, and then there's the DNA primase and the polymerases. Yeah, so that's the drawing for that, just to explain. And then you can see here you have a 5 to 3 going this way and a 5' to 3' going this way on that strand. So yeah, I think it's pretty self-explanatory for that picture.
Okay, and so for question 2, it's the vocab we found from the reading. So A says we got DNA primase. This protein complex synthesizes primers in eukaryotes. For B, we found that DNA A, this protein promotes the separation of the AT-rich sequences within the RIC. C, epsilon, this subunit of DNA polymerase 3 contains proofreading activity. For D, DNA polymerase alpha, this protein synthesizes the leading strand in eukaryotes. For E, telomerase, we were a little confused on this one, so we found telomerase in eukaryotes is the protein that displaces DNA polymerase 3 from the chromosome to terminate replication, and then TUS in prokaryotes. So we weren't sure exactly which answer we were looking for there. But for F, DNA polymerase 1, this protein has a 5 to 3' polymerase and a 5 to 3' exonuclease activity. For G, topoisomerase 2 or DNA gyrase, this protein introduces negative supercoils into the DNA ahead of the replication fork. For H, origin recognition complex ORC, and this protein binds directly to the ARS elements. And then for I, single-stranded RNA binding proteins, or SSBPs. And these proteins prevent DNA strands from annealing during replication. And for J, we found that DNA ligase, this enzyme seals Okazaki fragments into a continuous strand of DNA. And then I'm listing 3. So for A, it says list 5 enzymes, prokaryotic or eukaryotic, that consume ATP during DNA replication. And we found these also from the reading as well. And so we found helicase, gyrase, primase, ligase, and SSBPs do that. And then for B, list 5 enzymes, prokaryotic or eukaryotic, that consume DATP during DNA replication. And we found DNA polymerase 3, DNA polymerase alpha subunit, DNA polymerase beta subunit, also DNA polymerase epsilon subunit, and DNA polymerase 1 as well.
All right, for C, we have the list 5 enzymes, prokaryotic or eukaryotic, that have the 3 to 5 exonuclease activity. So we had DNA polymerase 3, DNA polymerase 1, telomerase, epsilon subunit, and the delta subunits. And then for D, indicate one enzyme, prokaryotic or eukaryotic, involved in replication that uses DNA as a template to synthesize RNA. So the DNA polymerase theta, and we had to do some outside research to find the answer to that one, to find what protein that was, or enzyme, excuse me. And then for E, indicate one enzyme, prokaryotic or eukaryotic, that uses RNA as a template to synthesize DNA. And this again is telomerase, and this we found in the reading as well.
Awesome. So I'll do number 4. I'm sure you all watched Sister Mayo's video. If not, you should, because she explains a lot better than I'm going to. But you have a template strand that's bold up here, and then you have a primer that's bolded right here. So I brought it down so I could visualize it, which makes it a little easier. You have the template strand on top, and then you have the primer on bottom. The primer, I had to flip backwards because it runs opposite of the template strand. So, and then it has different nucleotides floating around and basically says what kind of strand could form. So for A, running 5' to 3', you could basically just fill in the rest that's left after that primer because you have all the nucleotides that you need. So you have that full strand there. For B, you have I think it was dicyclic monophosphate, the di or cytosine monophosphate, but you didn't have enough cytosine, so you had to stop once you got to the first T. And as you can see, it goes TG, so it would go AC. So all you would have in that strand, 5' to 3, is an A. For C, you have U, so that's for RNA. So you don't have any thymine available. So running 5' to 3, you would just have AC because you hit that third where you would need a thymine, and they're not available. And for D, you have no guanine, so you would just go until you needed a G, and all you can do 5' to 3' is the ACT. And then the strand ends because it doesn't have any guanines.
All right, so for this one, we're given a list of proteins involved in replication and have to identify answer the question based on the protein. So for this one, which of the above proteins is found in a prokaryote? So we found that DNA gyrase, TUS, DNA A, DNA primase, DNA polymerase 1, and alpha subunit are found in prokaryotes. Now, which of the above proteins is found in the mouse musculus? So mice are eukaryotes, so we looked for all the proteins found in eukaryotes. So we have CDC6, the epsilon subunit, alpha subunit, pCNA, telomerase, DNA primase, and MCM helicase. And which of the above proteins is in chromosome compaction? We said DNA gyrase for this, and the reason for that is that DNA gyrase is involved with supercoiling, and so supercoiling compacts the DNA inside the chromosome. And that's where there was some confusion if chromosome compaction was the same thing as the DNA supercoiling. So we decided that, yes, they should be interchangeable based on our outside research as well. So which of the above proteins is involved in the initiation stage of DNA replication? So CDC6, DNA A, MCM helicase, and DNA primase. And which of the above proteins is involved in the termination stage of DNA replication? And the DNA gyrase, the TUS, and the telomerase. And again, most of these answers we found just from the readings and scouring those readings. So anything to add, Tianna or Keegan?
Nope. Looks good.
Thanks.
No, I don't have anything else. So if you guys have any questions, just comment below. Thank you.
Thanks.